Hello and welcome. Today, we are going to journey into a fascinating and somewhat unusual medical investigation. It is a story about a nutrient we all know, a vitamin we all need, but one that holds a secret risk that is incredibly difficult to spot. We are talking about vitamin A. But this isn't just about eating your carrots. This is about a group of researchers who embarked on a study to solve a critical diagnostic puzzle. They wanted to answer a question that has baffled scientists for years. How can we truly know if someone has too much vitamin A in their body without performing an invasive procedure? To find the answer, they had to look in a place most of us don't like to think about. They had to look at what happens at the very end of life to help the rest of us live healthier ones. Researchers are studying the complex relationship between what is floating in our blood and what is actually stored deep inside our organs. Specifically, this study aims to explore the connection between vitamin A levels in the blood and the actual reserves of vitamin A in the liver. You might assume that a simple blood test tells your doctor everything they need to know about your vitamin levels. If your blood numbers are normal, you are fine, right? Well, researchers are finding that when it comes to vitamin A, it might not be that simple. In fact, standard tests might be missing a hidden toxicity that is silently building up. So, what exactly is the issue? The study background highlights a significant gap in our medical knowledge. Minimal human data exists that directly compares the actual vitamin A stored in the liver with the biomarkers found in the blood. This is a massive blind spot. Why? Because the liver is the main storage unit for vitamin A. It holds the truth. The blood is just a transport highway, and the body is very good at keeping that highway looking normal, even when the storage unit is overflowing. This phenomenon is what researchers set out to investigate. Now, here is something that might surprise you. You might wonder, why don't we just check everyone's liver levels directly? The problem is that checking the liver requires a biopsy. That involves taking a piece of tissue from the organ. It is invasive, it carries risks, and it is certainly not something you can do for a routine checkup. So doctors have to rely on blood tests. But if the blood tests are not reflecting the reality of what is in the liver, we have a problem.
We could be missing cases of hypervitaminosis A, that is the medical term for vitamin A toxicity, until it is too late. This study was designed to bridge that gap. Researchers wanted to validate a specific blood biomarker. They were not looking at the standard vitamin A test you might be familiar with. They were looking for something more specific. They focused on something called retinol esters. The hypothesis was that the percentage of total vitamin A in the blood that is in the form of retinol esters could be the key. They suspected that if this specific form makes up more than 5 to 10% of the total, it might be a red flag that the liver is full. But to prove this, they needed samples, and this brings us to the most unique aspect of this study. How do you get matched blood and liver samples from human beings to test this theory? You cannot ethically ask healthy volunteers to undergo a liver biopsy just to test a hypothesis. So the investigators turned to autopsy samples. This study involved procuring matched serum, which is the clear part of the blood, and liver samples from 27 adult cadavers in the United States. These were donors aged 49 to 101 years old. It is a somber thought, but these donations provided a rare and valuable opportunity to see the biological reality that is usually hidden. Let's pause for a moment. Think about that age range, 49 to 101. These were adults who had lived full lives. By analyzing their tissues, researchers hope to understand how vitamin A accumulates over a lifetime. Were these individuals walking around with hidden toxicity? The researchers aim to compare the total liver vitamin A reserves with that specific blood marker we mentioned, the percent of total serum retinol as retinol esters. They wanted to see if that blood marker effectively spilled over when the liver was full. The process of analyzing these samples was incredibly meticulous. This was not a quick dipstick test. The study details describe using advanced chemical analysis. For the liver samples, they didn't just take a tiny snip. They extracted samples from every available lobe of the liver. Why? Because vitamin A might not be distributed evenly. They took 1 gram samples in triplicate from each lobe to ensure accuracy. Then they used a process called saponification. This involves heating the sample with a potassium hydroxide solution at 45 degrees Celsius for an hour.
It is a chemical breakdown that allows them to extract the retinol and measure it precisely. But wait, it gets even more precise.
After the chemical breakdown, they used a technique called ultra-high-performance liquid chromatography, or UPLC. This is a high-tech way of separating a mixture into its individual components. Imagine trying to separate a cup of coffee back into water, beans, and sugar. UPLC does that for chemical compounds. They injected tiny amounts, just 2 microliters, into the machine to measure the exact concentrations of vitamin A. They did this for both the liver samples and the blood samples. They also looked for other things. They analyzed the blood for markers of inflammation, like C-reactive protein. Why would they do that? Because inflammation can mess up vitamin A readings. The body reacts to stress and illness by changing how it moves vitamins around. By measuring inflammation, the researchers could be sure that what they were seeing was a true reflection of vitamin A status, and not just a temporary reaction to illness. So, what were they looking for? They were looking for a correlation. They wanted to see if they could draw a line on a graph, where as the liver stores went up, that specific retinol ester marker in the blood also went up. If they could find that link, it would mean that we finally have a non-invasive way to diagnose vitamin A toxicity in living patients. We wouldn't need biopsies. We would just need to look for that specific spillover in the blood. This is where the concept of homeostasis comes in. Even though the study doesn't use that word explicitly in the summary, it is the underlying principle. The body tries to keep blood levels stable, but the researchers suspected that when the liver, the storage tank, gets too full, it starts leaking a specific form of the vitamin back into the bloodstream. That leak is what they were trying to catch. The study suggests that a cutoff of 5% to 10% of total serum retinol being in the form of retinol esters could be the diagnostic line. If you are above that line, your liver might be overloaded. Now, you might be asking yourself, why does this matter? Why is vitamin A toxicity such a concern? Well, while this study focuses on the diagnostics, the implications are huge. We live in a world of fortified foods and supplements. We add vitamin A to milk, to cereals, to multivitamins. It is possible that some people are getting more than they realize.
And because the standard blood tests don't always show it, they might continue taking more, unknowingly adding to the burden on their liver. This study aims to give doctors a tool to see that invisible burden. The researchers collected data from 13 males and 14 females. It was a small group, but in the world of autopsy studies, this is a significant data set. Each person provided a unique snapshot of human biology. The researchers looked at the total liver vitamin A reserves. That is the gold standard. That is the truth of the matter. And they compared it to the percent total serum retinol as retinol esters. Imagine the precision required. They had to ensure the samples were preserved perfectly. Vitamin A is sensitive to light and air. If the samples degraded, the data would be useless. The study protocol mentions obtaining these samples through a request to the National Disease Research Interchange Service. This highlights the collaborative nature of medical science. It takes a network of organizations to make this kind of research possible. But here's a curiosity for you. We often think of malnutrition as a lack of vitamins. We worry about not getting enough. But this study flips that narrative. It suggests that for some populations, especially in countries like the US, where fortification is common, the opposite might be true. We might be dealing with a silent epidemic of too much of a good thing, and until now, we haven't had the right tools to see it. The researchers also looked at other carotenoids, things like lycopene and beta-carotene, using routine HPLC analysis. This gives a broader picture of the nutritional status of these individuals. It wasn't just about vitamin A in isolation. It was about the whole profile of these fat-soluble nutrients. Let's go back to the methodology for a second. The researchers didn't just look at one point in the liver. They looked at multiple lobes. This is crucial because the liver is a large, complex organ. If they had only sampled one spot, they might have missed the full picture. This attention to detail, extracting in triplicate, sampling from multiple areas, adds a layer of robustness to their findings. It means that when they say there is a correlation, we can be more confident that it is real. The study also mentions analyzing the samples for Hypervitaminosis A. This is the clinical condition of having toxic levels.
The fact that they were specifically evaluating this condition using autopsy samples suggests that they believe it is a condition that might be going undiagnosed in the general population. If standard tests miss it and we only find it at autopsy, that is a scary thought. It means people could be living with chronic toxicity symptoms, maybe vague aches, liver issues, or skin changes, without ever knowing the cause. Consider the outcome measures. They were looking for a direct statistical relationship. They wanted to be able to say, if your blood test shows X, your liver definitely contains Y. This kind of translational research is what moves medicine forward. It takes a biological fact, that the liver stores vitamin A, and turns it into a clinical tool, a blood test cutoff. But there is a catch. The study used cadavers. These were people who had died. Can we apply these findings directly to healthy, living people? That is always the question researchers have to ask. However, the biological processes of storage and blood transport are likely the same. The autopsy samples simply allowed the researchers to see the endpoint of those processes. The researchers also noted the importance of de-identified clinical history summaries. This means they didn't just have the tissues, they had some information about the donor's medical pasts without knowing who they were. This context helps them interpret the data. For example, did a donor have liver disease? Did they take supplements? While the public summary doesn't give us the individual stories, the researchers had access to this vital context to make sense of the numbers. Now, think about the future. If this study confirms that the retinol ester percentage is a reliable marker, how might that change your next doctor's visit? In the future, if you have unexplained symptoms, your doctor might order this specific test. They might look at that 5% to 10% cutoff. If you are above it, they might tell you to stop the multivitamins or to cut back on certain foods. It could prevent serious liver damage down the road. This study, conducted in the US, is a piece of a global puzzle. Vitamin A status varies wildly around the world. In some places, deficiency is the killer. In others, like potentially the US, toxicity is the ghost in the machine. By establishing a reliable biomarker, these researchers are giving global health experts a ruler that works for both ends of the spectrum.
It is fascinating to think that the key to understand this nutrient balance lies in the ratio of two different forms of the same vitamin, retinol versus retinol esters. One is the active currency, the other is the storage form. When the storage form starts flooding the currency exchange, the blood, that is when we know the bank is full. The study also involved checking for alpha-1 acid glycoprotein, another inflammation marker. By doubling up on inflammation checks, both CRP and this one, the researchers were being extra careful. They knew that inflammation creates a noise that can drown out the signal of vitamin A toxicity. They wanted to filter out that noise to hear the signal clearly. As we approach the end of this deep dive, remember that medical science is often about refining our vision. It is about building better lenses to see inside the human body. This study, with its careful collection of liver and serum samples, is one such lens. It challenges the status quo of standard blood testing. It suggests that we might need to look deeper, or at least look differently, to understand our true health status. Researchers are studying this because they want to keep us safe. They want to ensure that the vitamins we take to improve our health aren't secretly undermining it. It is a delicate balance, and studies like this help us find the center. So, the next time you see a label on a vitamin bottle, think about the complex journey that nutrient takes, from your mouth to your blood to the deep storage of your liver. And think about the researchers who are studying those 27 donors, using their legacy to ensure that we can detect the warning signs before it is too late. Thank you for joining me on this exploration of clinical research. It is a complex topic, but understanding the why and how behind these studies empowers us to understand our own health better.
Please like and subscribe my channel for more interesting videos.